All right, so thank you so much for the introduction. So I'm Malak, and today I'll be talking to you about how we are using multiple analytical methods to understand why certain plants become invasive. In particular, we are actually looking at how chemical biology via metabolomics can reveal the molecular tricks that invasive plants use to outcompete native plants. At the same time, I hope throughout the talk you could see how combining different multimodal approaches, each with its own strength and limitation, could give you a more complete picture of the chemical drivers of plant invasion, regardless of the challenges that comes with integrating multi-platform datasets. Just to start more general, as you all know, climate change is intensifying weather events, threatening ecosystem functions like nutrient cycling. But we all know at the same time, soil microbes and plants play vital roles in maintaining these ecosystem functions. However, soil microbes and plants, both are living organisms in any ecosystem, are also expected to be influenced by changing environmental conditions, therefore influencing ecosystem function. Now, to better understand ecosystems, we need to understand individual organisms. And to understand individual organisms, we need molecular level analysis. Only by examining molecular level processes through chemical biology, in this particular case, metabolomics, we could then gain crucial insights into how individual organisms will respond and adapt to environmental changes and how this influences ecosystem processes. Now, plant invasion is a common phenomenon that affects ecosystem biodiversity and is likely to intensify with future environmental changes. So we hope by understanding the molecular basis of invasion via chemical biology, we could then develop better strategies to protect ecosystems. So now our study site is the Santa Rita Experimental Range in Arizona. It provides a great example of how introduced species can dramatically alter native ecosystems. So roughly in 1930, lemon lovegrass, which is a warm season perennial bunch grass that is native to Africa, was introduced to Arizona to enhance soil stabilization as well as to provide food for cattle. However, since its introduction, this species has become the predominant perennial grass in the introduced area, outcompeting with native grasses and significantly reducing biodiversity. Now, this rapid and successful invasion highlighted a critical knowledge in our understanding of ecosystem processes. Now, despite observing the ecological consequences of such invasions by seeing that the biodiversity is decreasing tremendously, the underlying molecular mechanisms that enable certain species to thrive in new environments remain largely unknown. So our chemical biology approach that we implemented in this study aims to bridge this gap by investigating the molecular level processes that drive the invasion of this lemon lovegrass, potentially informing more effective management strategies and contributing to broader ecosystem preservation efforts in the face of both invasive species and climate change. So we went to the Santa Rita Experimental Range and we collected both soil and rhizosphere samples that belong to two different plant species. We had the lemon lovegrass, which we're going to refer to the invasive species, as you could see on top. Also, another way I refer to it is ERLE, which is the scientific name of this plant. We also collected root and rhizosphere samples belonging to the native community. In this case, we chose Arizona cotton top, or in other words, DECA. We collected again root and rhizosphere soil samples from these two plant species in triplicates from 2 different locations within that experimental range. We chose to collect samples that are located in open areas. They are exposed to the sun, and these areas are known to be low fertility areas. We also collected samples from plants growing under canopy, in this case, mesquite trees, and we wanted to see how these 2 plants grow when they are exposed to the sunlight and drought impacts throughout the day, and how they grow when they are under the plant, which is an area that's known to be high fertility area. With, again, the main goal is to understand the molecular mechanism that's allowing this plant to actually take over this experimental range. Now, for those of you who worked with chemical biology approaches or metabolomics, you realize that metabolomics is really plays a crucial role in understanding how plants invade ecosystems. So metabolomics is basically the study of small molecules that we refer to them as the chemical language of life. They mediate interactions between plants and the environment, including interactions between plants and microbes, as well as microbes and microbes. However, characterizing the diverse array of metabolites present in biological systems is really challenging because they're really diverse. And here I differentiate between primary metabolites and secondary metabolites. We know a lot about primary metabolites. These are metabolites involved directly in plant growth. These are sugars, amino acids. They are always produced in large concentrations, and we know a lot about them. What we don't know much about are secondary metabolites. These are organic compounds that are not involved directly in the growth of the organism, but rather they enhance the fitness of the organism. These could include alkaloids, these could include flavonoids, as well as antibiotics, all of these compounds that could be produced as a byproduct of a reaction, or the plant will only, or the microbe or the plant will only produce them under specific environmental disturbance. And that's why we don't know much about them. And even when they are produced in a small concentration. Considering the vast diversity of organic compounds present in nature, unfortunately, we don't have a single analytical tool or method that could allow us to detect and identify all the metabolites present in a complex biological sample. This limitation forces us to integrate multi-platform approaches. For this particular study, as you could see on the image here on the bottom, we chose to use 4 different approaches to be able to characterize the wide diversity of organic compounds in a sample. This is great, but also, as you're going to see later on, I'm going to talk about it at the end, it introduces a lot of challenges. But here, what I'm going to focus on this slide is to kind of give you a summary of why we chose these 4 approaches. We chose to start with FTICR-MS. This is high-resolution mass spec. It allows us to look at the bulk changes or bulk composition of organic compounds present in any sample. Then we complemented that with liquid chromatography mass spec because it will allow us to kind of identify the nature or the name of some of these metabolites. We then complemented LCMS and FTICR-MS with MALDI mass spec, which is imaging mass spec. And it's really powerful, especially when you're dealing with plant samples, because it allows you to look at the spatial distribution of these organic compounds in the tissue of the plant. And this has a lot of significance because depending on where that organic compound is present in a sample or in a tissue, it tells us a lot about how it's being stored and how it's being used. We then complemented all these mass spectrometry tools with NMR spectroscopy because we wanted to look at these small molecular weight compounds. So we believe by integrating the data from these 4 approaches, we could have a better idea about the type of metabolites present in our sample. I want to show you another figure to kind of further justify why we need those 4 tools, ignoring the fact that, as I mentioned earlier, each of these tools have their own strength and limitation. So what I'm showing here on the top is a histogram. On the x-axis, we have the molecular weight of the metabolites that we observed in the roots of the invasive species. And on the y-axis, we have the frequency, how many metabolites we've seen with that particular mass. And I want you to look at the different colors on this panel because each color represents the mass range distribution of the metabolites that was observed in that particular platform. For example, if you look at the green, these are the metabolites identified and characterized by NMR. And you could see that they capture metabolites which up to 300 Dalton. However, if you look at the liquid chromatography mass spec in blue, you could extend your mass range to a higher mass range. With FTIC RMS, we could also go higher up to 900 or 1000 Dalton. So this panel or this bar diagram shows you the importance of using different approaches because it allows us to capture a wide distribution of the metabolites in our sample. This is the example for the root of the invasive species. We could do the same thing for soil samples. Here I'm showing NMR, liquid chromatography mass spec, and FTIC RMS. And again, you could see how powerful it is when we use different multi-platforms to actually characterize organic compounds. Here I'm missing the MALDI mass spec or the imaging for the soil because it's not as advanced compared to imaging of root and tissue samples. But again, just to kind of show you that even though we know we expect to see a lot of challenges, the more data, even though it's always good to have a lot of data, there's so much challenge that comes with having multiple data, especially when you're trying to integrate it all together. But again, we do that because we know that by integrating different platforms, we could have a better idea of what's going on in our system. So now, leveraging all these platforms, we aim to kind of understand how root and rhizosphere metabolic profiles of the native and the invasive species differ from each other. And we predicted that the metabolic profile of the invasive species to be significantly different than that of the invasive species. So we knew that coming into this project, we expected that both of them is going to have different metabolic profile in the root, and this will be extended into the rhizosphere because we know that the rhizosphere is significantly impacted by root inputs. We were then hoping to take all that data and identify the molecular mechanisms contributing to the invasion success. Now we went back to the literature and we looked up a little bit more about what is known about the molecular mechanisms by which plant invade. And for this particular study, we focus on 3 mechanisms that are known in the literature, and we wanted to see if our multimodal approach here allows us to get more insight on whether this native species is outcompeted by the invasive species due to these 3 mechanisms. The first mechanism, we wanted to kind of investigate the nutrient acquisition by both plants. Usually it's known that invasive species have a more efficient uptake of resources, especially nitrogen. So we wanted to use our metabolomic data to see if there is any differences in the nitrogen uptake between these 2 plants. Second hypothesis or mechanism we wanted to test is called enemy invasion evasion, which is basically when an invasive plant thrives in a new environment because it has left behind its natural predators and pathogens, and therefore this plant does not have to invest a lot in producing antimicrobial compounds to fight infection, but rather it will invest in growing and competing with any other species because it have a lot of energy and resources to do that. And the third mechanism we wanted to test is what we refer to as phenotypic plasticity, which is an indication of stress tolerance. In other words, plants that are known to have these traits, it's because they are able to change what they do and their metabolic profile depending on the environmental condition. Basically, they've adapted to any changes in the environmental conditions, allowing them to change what they do to kind of overcome any of the stressors in that particular environment. Now, before we go in and talk in more details about how our metabolomic data validated these hypotheses or these mechanisms, let's take a step back and look a little bit at the overall distribution of the metabolites in the roots and the rhizosphere of these 2 plants, the native and the invasive species. So what I'm showing on this figure is an ordination analysis, in particular NMDS analysis or non-metric multidimensional scaling analysis. It allows us to look at the metabolic profile in each sample and see how it's similar or different to another samples. Here I'm showing you the data from 3 analytical platforms. We have FTICR on panel B, LCMS in the middle, and then NMR on panel D. Each circle represents one sample. Purple will be always representing the invasive species or ERLE throughout the presentation. Green will be the native species. Now, the open circles represent samples collected under open areas, whereas the closed circles represent samples collected from under canopy. Remember, our goal here is to look at the root metabolome of these 2 species under open areas and under the mesquite, as well as we're going to do the same thing for the rhizosphere. So now if you take a minute to look at these NMDS analysis, what you could see is that our hypothesis was validated by all 3 platforms, which tells us that the metabolome profile of the roots of native and invasive species appears to be significantly different, because you see a wide separation between the green circles and the purple circle, suggesting that yes, the root metabolome of invasive and native appear to be really different, even though they live in close proximity in nature. Now, the effect of the canopy comes next. So the plant species type is the major driver, followed with an effect of the canopy, which we're going to talk about the canopy effects later on. But for now, you could see that both of them have a completely different metabolome profile. Now, to understand a little bit which kind of metabolites are driving this separation, we could then overlay on this plot the different classes of compounds. And I'm going to show that I'm showing here the data only for the FTICR because it's easier to look at. But if you look at panel B, you could see that when it comes to the invasive species, which is in purple, we see higher abundance of amino sugars, proteins, and sugars throughout. And this high abundance of these labile compounds suggests that the invasive species are investing, or they have a lot of emphasis on growth and nutrient acquisition. On the other hand, if you look at the green dots on panel B, you can see that the native species are associated with a lot of lignin-like and phenolic compounds. Now, if you are a little bit familiar with lignin-like and phenolic compounds, usually plants produce a lot of these metabolites when they are actually under stress. Some of the phenolic compounds are known to be antimicrobial, and therefore plants could be producing them to kind of fight infection. Also, a lot of plants are known to produce phenolic compounds in the root to strengthen their cell wall to prevent water loss, especially if they are grown in an environment that is really dry with a lot of nutrient limitation. So this collectively shows you that the native species, rather than investing in growth and nutrient acquisition, they're spending a lot of energy and resources in defensive mechanisms because their root structures have a high abundance of lignin and phenolic compounds. So again, using this multi-platform method, we started to get some insights about how these 2 species are different. Now, this is what's happening in the root structure. We could then move on and see what's actually happening in the rhizosphere. When we look at the rhizosphere, now we see a little bit of differences here. If you look at the purple and the green, the open and the closed circles, you see that what is actually driving the separation is first the canopy type. You could see that the darker circles are separated from the open circles, suggesting that the major driver of the metabolites in the rhizosphere is being influenced on whether the plant is being grown under canopy versus closed area versus open areas. And second, we see that the type of the plant matters. So what this suggests that the rhizosphere metabolome is influenced by both the plant type and the environmental factors. Kind of you have an interactive effect here. Now we know that the plant type drives the type of the metabolome in the root. However, the canopy environment will then affect the rhizosphere by modulating root exudation. So in other words, the rhizosphere here is being influenced by the canopy first and then by the plant type, and therefore this reflects that the plant traits and its response to the environment through changes in the root exudates is what is controlling the rhizosphere. Okay, so again, you could see that all three analytical platforms showed the same effect. So the roots are directly influenced by the plant types. The rhizosphere is being influenced by how the plant types are responding to the environmental conditions, thereby changing what they release below ground, leading to an effect in the rhizosphere. So this kind of tells us a little bit about how the metabolic profile changes between those two native species. Next, we were really interested in then seeing if we could actually use that data to kind of validate some of the mechanisms that we've seen in the literature. So that's kind of the question I'm asking here. Can we use metabolomics data to infer mechanisms? So what I'm going to be discussing first is the first mechanism, which is the nutrient acquisition strategies, focusing on nitrogen. And the reason I focus on nitrogen here is because these plants are growing in a semi-arid ecosystem that is really limited or nutrient poor, and thereby we wanted to see if these two plants are behaving differently. Another thing I want to point out here is that if you take plant tissues from the invasive species and look at the carbon to nitrogen ratio, you could see that the plant tissue of the invasive species have a low C to N ratio, or in other words, have much nitrogen in its biomass, and that's why cattle will prefer that. So we wanted to understand why these plants have a higher nitrogen ratio and how this influences their ability to compete or invade. So what I'm showing first are the percentage of nitrogen-containing compounds for both the invasive and the native. Again, the invasive is in purple or ERLE, and the native is DECA. And here I'm using FTICR data. Just want to look at the overall changes in nitrogen-containing compound. As you can see from this data, that we have a much higher proportion of nitrogen-containing compounds in the roots of the invasive species, which again, they are in purple, compared to the native species in green, which is consistent with the fact that the biomass of this plant has much more nitrogen compounds, which is good kind of correlation between C to N ratio and the FTICR data. So if you have more nitrogen content based on just an total nitrogen analyzer, we're seeing from the FTICR data, we have a higher abundance of nitrogen-containing compounds. Now, what we've done next was to then use our LCMS, NMR data, and imaging mass spec to kind of investigate more the nature and the localization of these nitrogen-containing compounds. So first, we focus on organic compounds that are known to be involved in nitrogen assimilation. And here I'm showing kind of a pathway of all the different nitrogen-containing compounds we detected in our combined LCMS, NMR, MALDI, and so on. And I'm looking at how the abundance of these compounds changes between the native and the invasive species. So overall, when we looked at these compounds, we saw that the native species in general have a higher abundance of compounds involved in assimilation. And this kind of suggests that the native species have an efficient nitrogen use strategy compared to the invasive species. So what does this mean is that those native plants, whenever they get a hold of nitrogen, they quickly incorporate nitrogen in its tissue. And that adaptation is known to be beneficial in environments where nitrogen is limited or competition is high. So that's when it comes to the native species, that's what they're doing. Interestingly, when we now looked at the invasive species, we focused on something we call nitrogen carriers. So nitrogen carriers is basically organic compounds that facilitate the transport or transfer of nitrogen atom or groups between different chemical species in a biological or a chemical process. I'm showing on the right the different compounds that are known to be nitrogen carriers that we identified with liquid chromatography mass spec, and the abundance of these compounds. And overall, if you look at these data, I'm only showing a subset of these nitrogen carriers, you could see that the majority of the nitrogen carriers we detected in our samples seem to be more upregulated in the invasive species compared to the native species. This high regulation suggests that the invasive species focus on effective nitrogen acquisition and mobilization, enabling more efficient uptake from the soil, storage for later use, or transport within plant compared to the native species. And this is really important, again, when you are competing with other plants, you are in a nutrient-limited environment, so you're way better in taking that nitrogen from the soil and moving it within the plant tissue or storing it for later. And this will tell us that our metabolomics data so far validate the hypothesis that invasive species have a more effective nutrient acquisition, and that's why they tend to invade, especially in environments where we have a lot of competition because we don't have much nitrogen to start with. Now, what we've done next is we said, okay, great, we saw a lot of nitrogen carriers in the LC-MS data. Can we now go back to our imaging mass spec data, find these metabolites and see their localization within the root structure? We were able to actually find all these 3 metabolites, which is great to see that most of these techniques kind of corroborate each other. But also we were able to find additional nitrogen carriers that the MALDI mass spec was able to detect, but we did not see them with the LC-MS. Here I am showing the MALDI imaging of these same 3 compounds. And on the left, I'm showing the abundance of these compounds from the MALDI mass spec. And on the right, far right, I'm showing the abundance of these compounds using the liquid chromatography mass spec. It was good to see nice consistency between those 2 completely different approaches. Sample prep is completely different. With liquid chromatography mass spec, which is on the far right, we have to extract the metabolite using an organic solvent. Whereas I know Dushan gave you a presentation, I think yesterday, about MALDI mass spec. It had a completely different sample prep. So it was great to see that we detected the same compounds with both approaches, and the abundance of these metabolites follow the same trend. Now, if we focus on the middle panel, here I am showing an image of the localization of this metabolite within the root structure. I'm showing the MALDI, where you could see all these yellow and blue colors, where you could see like the yellower it is, the more abundant that metabolite. And then I'm showing the microscopy image on how the root looked like. This is not the best microscopy images just because these roots are really thin. So Dushan and his team had a very hard time trying to dissect these roots. But overall, we were able to get some really good data. Now, what I want to point out here is the localization of these metabolites. As you could see that we find these metabolites with both plants. However, they tend to be way more abundant in the invasive species compared to the native species. If we focus on the metabolite on the bottom, which is glutamine, you could see it's present throughout the whole root structure. However, if you look at the one on the top, asparagine, which seems to be the most dominant nitrogen carrier, it's very close to the xylem of the plant root, which is basically suggesting that it's being used for transport within the plant. So again, using this multimodal approach, we were able to validate that we have a lot of nitrogen carriers in the invasive species root, suggesting that this invasive species have very superior or efficient nitrogen acquisition and mobilization within the plant that makes it superior to the native species and helping it out compete, especially, again, we are in an environment, we don't have much nitrogen resources. Okay. Now, this is the nitrogen story. Next, we wanted to see another or validate another mechanism, which we refer to as phenotypic plasticity. So again, as I mentioned, phenotypic plasticity is the ability of a plant or an organism to kind of change or modulate their metabolome profile based on the environmental conditions that they live in. If the plant or organism are capable of doing this, then they have a better chance of surviving and competing with native plant. And our hypothesis coming into this well, the invasive plant should have a higher phenotypic plasticity trait compared to the native species. And that's what we wanted to use our metabolomic data to validate. So here I'm showing a lot of figures on this graph, and I'm going to walk you through that. But basically, we now use the data from FTICR, LCMS, and NMR to kind of investigate the phenotypic plasticity mechanism. So let's start first with panel A. Panel A, here I'm showing overall the changes in the metabolite diversity between open and closed areas, between invasive and native species. As you could clearly see, just when it comes to the metabolite diversity, we did not see any impact. It seems that the diversity or high diversity of metabolite seems to be relatively the same between both, and we don't see any effect of open versus closed. But this is, again, just metabolite diversity. It doesn't say anything about whether it's capable of changing what it's doing or not. So to kind of further dig into that data, let's look at panel number B. So what we're doing on panel number B is basically we are looking at the metabolites that we observed for each plant under closed areas and under open areas. And the goal here is to see how much metabolites were shared if the plant was growing under open and closed conditions. So the white circle represents how many metabolites were shared, whereas the blue and the yellow is how many was unique under a specific condition. For example, if we look at the invasive species, which is in purple, there was 3,662 metabolites shared on the root if the plant was grown under open areas or under closed areas. If it's grown under closed areas or canopy, around 628 metabolites were unique versus 504. Now, if you look at the native species, which is in green.
Alok, can you wrap this up, please? We are kind of running out of time.
Okay, sounds good. So overall, what I'm trying to show here is that when we look at both plant species, we see a much more amount of metabolites shared in the native species compared to the invasive species, suggesting that invasive species actually change what they're doing depending if they are grown under open areas and closed areas. This is with the root metabolome. And then quickly here, we're seeing the same thing in the rhizosphere. And I want you, just look at the bottom figure when it says 2 ERLE open versus closed. We could see that the invasive species really changes what it's doing in the rhizosphere when it's grown under open versus closed condition, again, suggesting that this invasive species have a lot of phenotypic plasticity. And the final mechanism I want to talk about is the defensive strategies. As you've seen earlier, that these plants have a lot of, the native species produce a lot of phenolic compounds. As we've seen earlier, we dug into the LCMS and NMR data, and we actually confirmed that most of these phenolic compounds we are seeing are antimicrobial. We then grouped all these data together, as I'm showing on the panel on the bottom. And again, you can see all that green suggests there's so much production of antimicrobial compounds by the native species, suggesting that they're investing so much energy in fighting infection, whereas the invasive species is actually happier and just investing in growth. So quickly, to just sum up, I'm going to skip this part. To just sum up, we use multimodal approaches to validate that the invasive species employs superior strategies or traits that allows it to compete. And this brings me to the quick last slide, which is related to this workshop, is that integrating all this data was great. It gave us a comprehensive look into plant invasion mechanism. However, because we had 4 different approaches, each approach is different, samples are prepared differently, the instruments work differently, it had a lot of challenges. For example, when it comes to chemical imaging, it was really time consuming to actually look at each image and look at the distribution of the metabolite within that image. And this is also subjective to the user. Also, we had, while I did not present all the data, we had some inconsistencies between MALDI and liquid chromatography mass spec, even though in certain cases they were both corroborating each other. At other times, we had inconsistency, but also just integrating different data sets poses a lot of computational challenges. However, we are aware that we need to integrate these approaches because there's so many advantages of integrating these approaches. So moving forward, just a few points for everybody to think about is that we need to kind of do a little bit better in kind of helping each other to better integrate different data sets to kind of better see what's going on in our system. With that, I would like to thank you all for listening and for giving me the opportunity to share some of this work. Thank you.